Second, we're muted here. Unmuted.
I can hear you.
Okay. So welcome to day 3 of workshop 29 for 2022 May, the demultiplexing workshop. And so today's our last day, and I don't think it's going to be a full day. Let's see. Go to full screen here. Okay. So I'm going to delete a few things I thought I was going to cover. Phi-X, we don't really spike it in anymore, and take some time to get in, explain what it is and why we did it and so on. So I'm just going to, since it's less of an issue now that we don't do it anymore, I just drop it. And looking at the problems and trying to give detailed examples for all the kinds of problems with actual real lane examples, it would take forever. And so I'll just give you the kind of the list of problems, but we're not, I don't think really going to get into examples of each one and how you would actually detect and remediate each one. You already kind of know a little bit about how to do histograms and stuff. And sort of like, if you got a lane in front of you that's a problem, then you can start to dig in and figure out what's your big situation rather than sort of telling you them all before they ever happen to you. So I think that'll be the approach instead. And then if you're taking it for credit, there's a quiz and homework that I'll email out after the workshop's done. We always recommend everybody do those. Okay, so we ended up last time with index hopping, and I forgot to include one of really the example lane that really we did very early on in the Nova feed, and was the lane that really convinced us this was really seriously a problem. This was a 448 plex, very large number of samples in a 2/3 full non-unique dual, with the pattern on the right there being the pattern that was expected. So the white means no sample expected, and the brown means sample expected. But when you look at the actual results, the squares that are white aren't white. Now they have this right-hand half or whatever that's sort of like blue, which is basically a zero log scale color scheme, of course. But this was a case where like there were squares that were on this log color scale orange now, right? I mean, and so it turns out about 12% of all the reads that go to user indices land in unexpected places. So this was a case where actually there was stuff we could do in it. It turned out that the prep needed to be worked on, which you can kind of guess because this shows a block structure, right? So it's going to use that sort of a script that blue and the comparison there's something happening related to it. It's not just an all versus all. So indeed, there was a way to sort of clean up the prep a little. And we made this problem more than 10-fold less of a problem, maybe even 100-fold less of a problem with some tweaks to the prep. Okay, so the next topic is duplicates. Many sequencing experiments result in duplicates. And by duplicates, we generally mean that there are two wells on the flow cell that sequence all of the reads. Single end, the main single end read, of course, which you always have. If you did paired end, then we also include the paired end in that list, and then also the two indices as well. So basically, whatever, if you did a full paired end and don't do index lane, we're talking 4 reads for the well, agree with the 4 reads for another well. There can be lots of reasons for that. It's not like instantaneously good or bad. It's just a thing, right? It's just a duplicate. It's just you get the same information on the 2 wells. And usually when because there are base call errors and so on, usually in full generality, you also have to allow some slop in that comparison. It's not necessarily, you're going to, you should, it depends on kind of whether you're biased towards like false, if you want a low false negative rate, if you want a low false positive rate. So if you are really worried about dupes and you're very, worried about false negatives, then you need to allow more slop in the comparison, right? Because you want sensitivity. So if the 2 guys are really duplicates, but one guy's got a base call error, they're not going to literally match that sequences. So you got to allow some slop in the comparison. On the other hand, if you're really worried about false positives, of course, as you take the slop higher, the chance of a false positive goes up. So if you're really worried about false positives, then your value tolerance is back down, maybe all the way to that. It kind of just depends on what you're doing. And different downstream applications are more or less sensitive to and so on. And different library preps have orders of magnitude variations in terms of how many groups they make. And sometimes the biology is the question, and it's something you want because it's part of the biology, as opposed to sometimes the dupes are from not the biology, and then that's what the kind of the case we really worry about. But anyway, so yeah, this comparison generally has to allow swap, at least in the general case, and that is one of the things that makes these hard to deal with, because if you haven't done anything else, you're very early in the processing of the lane, you haven't done alignment template, maybe you're not even in a situation where alignments are a thing you're doing. I mean, many, so the great divide in sequence applications is probably the methods that go align reads to a reference of some kind, reference genome of some kind, and then there's all the ones that don't. So that's sort of the first huge divide in terms of what path you're going to go down. The guys that go down the alignment to reference path have an easier way to do it. Because if you don't have that, then the other path, or even before alignment in that path, how are you going to detect duplicate? At this point, it's now an all versus all sequence alignment problem with a billion reads, right? So that makes it computationally challenging to do that all versus all comparison. Generally, to do that, if you don't allow swap, it's not a big deal. If you're doing exact method only, it's not that bad. If you allow swap, you're doing an all versus all billion against the billion, and it's then it's a little bit. And generally, the only way to make that fast is to be very large enough to go some sort of fancy to get structure. And sort of, if you can, just finding the pairs of sequences, or the, it's more than just pairs. When we say duplicate, when we, these two wells may have the exact same sequence. Well, let's just go to the exact same. This well and this well now have, let's say we're in pair A. So we've got 4 reads. So this guy and this guy have the same exact sequence of the read. That doesn't mean that's the only 2 copies. There could be another well over here that's the same as these 2 wells. So when we talk about duplicates, we usually consider them in groups, right? Which is all the copies that you consider sequence similar enough to be the same, right? So a group, some guy may have 1327 duplicates or something, right? And if it's exact matches, then those sequences are all literally identical. They're allowing swap, and they're all in some fuzzy ball. Maybe there's some kind of center consensus sequence or something. The problem of identifying groups then becomes not only you have to allow some swap in the comparison, but you essentially also have to do some kind of clustering where you're now deciding on consensus sequences, where you're going to draw the boundaries. If A is similar to B, with swap, if A is similar to B, is similar to C, is similar to D, is similar to D, is similar to F, by the time you get to A to F, maybe not similar anymore. So, there's these issues of clustering then and so on. It's not really straightforward once you bring swap into it. So yeah, so this is kind of the start of kind of like the messiness of trying to deal with dupes. The problem why they're a concern is that, well, if they come from the biology, they come from biology, okay, and in that case we want to keep them. Because any distortion of that would be distorting the actual data. But then there's the other kinds which are artificial, either from the library type or the instance. And those you worry about more because many sequencing applications have an assumption that different reads are independent pieces of that. So think about something like GHCK, genotyping, variant detection, SNP detection, genotyping. Often done with fairly sufficient coverage. You may have an average coverage of 4 or something. You really would rather genotype many samples than few. And it's only a diploid organism, so by the time you've seen, you've seen one observation, you've actually already, you've got to, well, that's a problem because you don't really know if it's homozygous or not yet. But if you see, say, 4 observations, you make a pretty good guess that it's either homozygous one thing, homozygous another, or heterozygous. So typically, GHCK genotyping is doing analysis of other left-handed organisms is very well known. You don't really have to make discovery, you're just doing genotyping. You may be running a barely sufficient coverage. And then duplicates could become a real problem because even small duplicates of a particular locus could change your base your idea of the genotype. Because you're assuming that each read is an independent observation. And anything that counts is also a problem, right? And lots of things count: RNA-seq, ChIP-seq, BS-seq, especially whole genome bisulfite BS-seq, because that tends to have low coverage. High-seq, anything that counts, obviously the copy number of evidence matters because that's the basis of the signal. Now, those degrade a little more gracefully than like GATK does, because as the dupe rate goes from low to high, the effect goes from low to high. It doesn't just instantaneously kind of reach a bad level as fast as GATK does. But, you know, and it kind of depends. You know, say like RNA-seq could be less sensitive because in RNA-seq, the loci, which are genes or whatever, transcript, you know, they have orders of magnitude variation. You know, what's gene expression bottom? 6 orders of magnitude, 7, 8, you know, whatever. It's a lot, and usually the lowest frequency guys, you know, the lowest expression guys, you can't do very good difference in expression anyway. So they're not really much of a focus. So as long as the, you know, as long as the dupes are hitting everybody kind of equally, and so on, then, you know, for a moderate to high expression guy, it's not that big a deal. Instead of getting 1,000 reads, you get 1,100, but you're comparing it to another guy that gets 2,000, which goes to 2,200. I mean, once the numbers get big enough, sort of the discrete nature of the duplication, it doesn't matter. The average rate kicks over, and that's probably more or less. So that's, you know, so anyway, so it's I just want to give you some appreciation that so the impact varies, and so it kind of depends on what you're doing. All right, so let's talk about the different sources where these things can come from. So yes, so they could be really in the original material, and some library preps are expected to generate huge numbers of dupes. For example, take like say RRBS, reduced representation by sulfate. That is a way to increase to not, that's a, you know, say you're in the human, which is a big genome, very expensive to so given that, people usually, you know, one choice is to trade away coverage on the whole genome and concentrate the coverage on a small fraction of the genome. That fraction of the genome typically being defined by a restriction enzyme. So you use a restriction enzyme, and now all of your reads start at places where that restriction enzyme cuts. Well, now you pass a very, you know, relatively small number of subsets. It's not like you're getting reads uniformly distributed over the genome; they're all piling at restriction sites. And so, and then the methylation state, which is reflected in the read sequence, is going to be the same for a lot of those reads. And so you synchronize the location, and the base data probably has a lot, you know, CD methylation, something that's mostly on or off. So like all cells, all copies are all CD, or all methylated Cs are all not, which is reads going to be C to P. And so you do expect a lot of literal duplication of data because that's the way the protocol is designed. And you wouldn't want to remove all duplicates, like take all groups of duplicates and each replace them by a single representative. That would be a story. And even things like RNA-seq could easily produce a fair number of duplicates because, you know, your transcripts, again, you know, some of them are expressed a million times more than others. And then you've got maybe poly-A purification or something. You end up with a 3' bias, right? So you're pulling on the 3' end. And you've got these orders of magnitude variation coverage. And so yeah, some of them are you're gonna have a whole, you know, for the moderate, you know, high expression, you're gonna have piles of reads that all start the same place. So yeah, so some protocols generate it, and you don't want to remove duplicates just with a sledgehammer and just kind of kill them all because that's not the right thing to do. It's going to distort those guys. So on the other hand, some protocols like whole genome bisulfite, all the bisulfite protocols in general say they usually have problems with sample quality and diversity because the bisulfite is very harsh and sort of by the time you kind of get all the samples prepared, there may not be kind of enough productive molecules left. And then when you say reach the PCR stage library prep, now you're amplifying differences in guys about their preferences. You may end up with an amplified library that's really not uniform. And so, so some of these protocols can generate them at appreciable numbers which are not reflective of biology. So like that could be 20, 30% of the reads could be artificially created by the PCR process. They've amplified some things have amplified, the other guys got lost, and so you end up with sort of a subsample from the original thing that's even kind of replacing. Not like independently distributed units without you're actually kind of just repeating and getting copies of the guys who survived. And so that's not good because then those don't reflect any kind of observations. And because the frequency is high, then you got to do something. Right? There's a point where sort of the dupe rate gets high enough, and then you kind of got to do something. So we'd like to remove these PCR dupes. And by remove, that's also kind of a tricky business because it doesn't really just mean, say, replace each group of wells that are the same by the same with a single copy. What you really need to do is kind of like have some way of estimating how many pre-PCR copies are appropriate. Kind of, if this sequencing run that you did was sort of magically deduplicated, how many copies really would have derived from the original material? And if you've got n copies that showed up and n copies are appropriate, you need to delete n minus n guys. Right? It's not like you should just kill all the groups besides one. So that's complicated because that depends upon estimating rates and sequencing depth and all kinds of things that affect that. And so many dupe tools are really not that sophisticated, and they don't come anywhere close to approaching that idea. And so the process of deduping is also introducing biases. So this is a serious problem because at the moment, with the information we have, we can't really separate the PCR guys from the true guys. We're going to need something else to help us make that decision because the sequences themselves don't come labeled with where they came from. So, you have a question? Say single-ended argument. You really don't want to just take sequences that are literally flashing then you very much expect to have repeated copies of that thing. And with the single-ended guys, you really don't have to do any progress. But actually, so okay, we've got a couple more sources before we talk about how we're going to deal with this problem. So there's PCR is usually the big source. And usually it's orders of magnitude. If you look at all the library preps that people do, some of them are more prone to it and not or whatever, but if you just look at the spectrum of the universe of all sequencing, the PCR duplicates are the ones that swing over the skew thing. When they're bad, they're really bad. So they're usually the ones that if you're worried about dupes, those are the guys that are the ones that are making you worry. So there are ones that actually come from the platform, from the machine. So in the old days, when the clusters were randomly generated on the flow cell was an oligo line. You flow your sample in, at the beginning of the experiment. Your individual prepared library molecules are randomly landing on that line. Bridge amplification is filling up the clusters for them. Bridge amplification is completely controlled, so some clusters are a little bigger than others. But in particular, also, there's nothing that stops them from overlapping. And essentially, that was the definition of PF1 versus PF0 in the random cluster data. PF0s, in terms of empty wells, didn't exist because empty areas of flow cells weren't called by the base the image analysis as a thing. Right? If the spot is just black for the whole experiment, you don't bother to even track those pixels. But one of the first steps of image analysis in that world is to decide which pixels belong together and to separate groups of pixels into different spots. And in that case, everything was a something. There was no empty space that wasn't even recorded. But the PF1 versus PF0 decision became our clusters overlap. So if you have two clusters that are overlapped, then you can't really separate the intensity signal very well. And so you have to fix the sequence code. And the PF0 would be indicating that the pipeline project and the two overlap. But the opposite side of that is over-segmentation. So that is where the image analysis stage at the time we're studying which pixels belong together in the same spot takes a spot that's really a single spot. Maybe it's unusually large or lumpy or something, and calls it in analysis two separate spots. But it's really the same spot. And so those two spots are going to end up calling basically the same base column. But they don't represent two independent pieces of evidence. They're just a single spot being called twice because it was broken up into two pieces. And so they call those optical. And it wasn't very commonly done to deal with them, mostly because, in our experience, the frequency was not. We're talking parts. So not everything is parts. Really, like low, single digit parts. There was, like in GHEK, which are probably one of the first people to really learn about this, loads that they're operating in. Their Picard has this mark. And that is actually one that depends upon alignment to reference. I didn't finish that discussion. We talked about the all versus all comparison. Well, if you do have alignments to reference, that simplifies your life a lot, because then you don't have to compare all to all. You just walk along the genome and take all the reads that are in a little vicinity around where you are in the genome. That's your candidate for comparing it. You don't have to do all of it. Things that map to nearby on the genome. Right? You go from a billion by a billion columns, lots of little dozen by dozen columns. The number of comparisons is really square on the side. You cut the same total number of guys, 12 by 12. Totally much smaller. Essentially, you're taking a gigantic thing that's a full grid and putting little squares down the diagonal, and adding the size of the stuff with a little square. So people did not, even the GHD tables didn't always, it was not too common. But you could do it. And there was some simplification there. You didn't really have to do an all versus all comparison, because the image analysis always went file by file. And so you knew that if two spots were in different files, it's impossible for the image analysis to kind of basically correctly segment it across that boundary. I mean, you have edge wells that could maybe, image tiles are really perfectly aligned with the end of one pixel at the beginning of the next layer. You have the edge cases. Edge cases are it's like a linear thing, but it's fraction cases. Like I say, the PCR problem is usually ordinally bigger. You're learning about trying to do this step, and then the edge cases of the file are like another little bit. So yeah, anyway, if you weren't gonna, it wasn't really a big deal to worry about how. Yeah, so that's the next slide. So XAMPP has duplicates too, but the mechanism is completely different. So with XAMPP, with pattern flow cells, once in a while, a DNA molecule in a nanowell can escape the well. So if it escapes, one of two things happen. And usually the first thing that happens is the plant is nearby. It sort of like crawls over the top and it goes into a well nearby. That's more of an isotropic process for sort of like any well in kind of the vicinity, in particular direction, kind of probable. But that falls off extremely sharply. The other thing that can happen is if the molecule can actually get in the z direction high enough off the surface, it can start to encounter the fluid flow. So because this is happening during XAMPP sample load, your library has been diluted and it's now flowing from one end of the flow to the other in the wrong direction. And so there is a laminar fluid flow occurring in this operation. And if you get high enough off the surface of the flow cell, you can join the laminar flow and take it to rise for a while. You come down and you crawl into a nanowell, or you flow and go into a nanowell, and that nanowell isn't already polymerized by somebody, you can start the polymerization reaction in that well. So you can end up with sort of these, if you sort of histogram the duplications of this type over entire flow cells, and you sort of bring them all back to the point where you sort of like have the originating, what you think is the originating spot down here at the bottom center. The pattern that you get is sort of a little circular ball, really tight around the beginning, and then an elongated comet-shaped tail that is the fluid flow. And it can, of course, be a chain reaction, right? Of course, it gets more, extremely fast. It gets rare, rare, pulse, and then it gets rare. So you have to happen twice as rare, but you can't theoretically. You do observe it once in a while. You can get two duplicates out of this well. You can get a well that lands to another well, which then another back in his case, and you kind of get these chains. In fact, if you use those changes in intelligent ways, that's what allows you to actually decide that the duplicates actually occur in the fluid flow direction. Basically, okay, so even detecting these is kind of a pain because essentially if you want to do this numerically, I'm going to detect all this wall comparison again. You can't really simplify it very much because the distance, the number of wells that are sort of in the zone of flow is pretty big. It's still like a small fraction of the flow cell, but it's not like a negligible fraction of the flow cell. In terms of the number of wells in the flow field, it's big. So it's mostly doing all this with all. It's very large amounts of memory, which you never have to do, especially so I did write in the Heising 4000, first pattern profile, I did write a tool with the large RAM tool, like maybe 40 gigs of RAM, maybe depends on the length, and then you obviously get out to do this thing. And we did it for dozens of runs. We never really saw very because basically, if duplicates are all a worry for you, you're going to be worried about PCR duplicates, which are orders of magnitude more. And so if duplicates are a problem for you, you're worrying about PCR duplicates, and you're going to have to be doing something to take care of those. And when you take care of those, you'll take care of these. And so there's not really much of a need to cut these independently, because protocols that have do problems and do things to correct it are going to take care of these as side effects. And so that's why you don't really see tools and sort of people doing just platform-based. We need to take care of all the fake dupe problems, including PCR, not just it. And that requires something. The way you do that will take care of the problem as well. So yeah, so this is basically the summary. The greens we want to keep, those are the biological true ones. And then the red ones we want to not just remove, but thin to the correct level of representation. So that means the PCR guys and the XAMP groups. The PCR ones are certainly confounded with the true dupes. There's not really anything we've talked about yet that would allow you to ever tell those apart. Of course, the XAMP dupes, you have some hope of sort of getting at least a probability that it's more likely to be XAMP because you have the physical proximity on the flow cell where, maybe if a flow cell is this big, maybe the XAMP cells are like this big. So if you get duplication sequences and they show enrichment for small distances on the flow cell, maybe with a little more allowance for distance in the flow direction, then those are probably XAMP. But anyway, it's not like that's like 100% reliable either decision. It's a probabilistic decision. Okay, so what do people do in terms of removing them or thinning them or whatever? Well, one thing people used to do, which is really not a great idea in many applications because it's potentially very quite harmful, is what I call the sledgehammer, which is to just literally take, say you got single-ended reads, just find, sort all the reads by sequence, and then just cross, just take each group that's the same and just keep one. That's a real heavy hammer. So that has the advantage of being probably pretty sensitive because the default assumption is base colors. Usually this is stuff people did early on, mostly when single-ended reads were more common, and at that time single-ended reads were shorter. So 50 mers. Generic 50 mer probably has no base color, at least the majority. So anyway, so this has some sensitivity, and it certainly has a low false negative rate, but it's probably way overkill. It's probably removing a lot of information that's not supposed to be removed. Now, if you can get to paired end reads, you got a chance of doing much better. Because when you get to paired end reads and you ask that the paired read is also a match to the paired read of the other guy, that actually implies that the insert length also matches. So if you think about the, we have the cartoon picture of libraries in day one, where it was adapter and then insert and then adapter. So we call it insert length, the length of the piece between the adapter. And you can't control that to base accuracy in library text. Right? Nobody, it's like, okay, your adapters are 42 bases. I don't know what they're, there's dozens. They're in the 50 range, something like that. You put it together. Let's just use round numbers, 50 mer adapters. It's not like when you finish library prep, all of your prep molecules are 50 plus 100 plus 50, precisely 200 bases long. You might have an average of 200, but you're going to have a distribution around 200. Something like this. So this is like a picture you'd get out of the bioanalyzer or something like that. So, here's the length of the DNA that's quantified in your library. So maybe the peaks at 300, maybe the adapters are 100, 200. But there's a spread, right? So I mean, it's this one kind of drops off by 200 bases and goes out to 600. There's some asymmetry on that distribution. But all these lengths are represented. I mean, it's not like it's not like a flat and a uniform flat, but it's like there's a fun. A lot of it would be between like 250 and 250, which is still 100 nucleotides range of appreciable variation. So if you were to have a dupe, if it's a true dupe, meaning independent molecules, then it has a chance to kind of draw twice from this distribution. And the chances that they both come out exactly at, say, 297 is kind of low. Because you got this 100 mer range that's kind of available with frequent hyper. But if it's a PCR dupe or an XAMP dupe, the insert length will be exactly the same. Because when PCR or XAMP duplicates this molecule, it's going to duplicate from adapter through the other adapter. And the whole construct will be exactly the same. So the insert length will be exactly the same. Whereas the true guys vary. Now it's not perfect, but that's actually good, really good, because that gives us our first way to sort of start to separate true dupes from fake dupes. It also handles the platform and PCRs equally. So that's what people do. And essentially, if you just ask the sequence match on the main end one and sequence match on the paired end, that is a check on this insert, because probably your sequence is diverse enough that if I slide you a couple of nucleotides forward and back, the sequence changes. Now, you could be in a region of puzzle polymers, but if the genome has 1,000 T's in a row or something, you're not going to, when you slide around, you're not going to know where you are in those T's. So most genomes, if I slide you around 100 plus or minus 100, I point to anywhere in the genome and slide around 100 bases in the window and all those windows. So it's not perfect, but that's great. So what that means is that when you do paired end removal, and even if you're only matching sequences, you got a chance that you're actually doing the right thing, because the trues have the spread, and that is reflected in even if the first end is matching, the second end is going to vary based on that insert. So see that. So it's basically observable variation. The important point is the insert length thing is observable variation that true guys vary in and false guys repeat exactly. It's not changing. And that's our mechanism by which to do the separation. Now, it's still a probabilistic separation. These are perfect. For example, as the tightness of this distribution cleans up, it works less well. And in particular, if the dupe rate rises, it works less well. Right? So if the typical size of the dupe group is 2, that means you're only comparing 2 draws from this distribution. If the typical size of the dupe group is 1,000, I'm going to have more, random, values of property. So it's not perfect, but that gives us the idea. Now, if you use QC tools like FastQC and FastP, and they tell you like your name is duplicated at some rate or something, or FastP, I think there's an author to deduplicate it for you. You've got to dig into documentation for what they're actually doing, because they're usually doing something that's not racist. Whether it's appropriate for your situation or not, could be yes or no. You'll take these tools, if you compare them against each other, they'll get orders of magnitude for you. It's not, it doesn't mean anything until you know what they're computing and how it relates to the situation. So just you know, be aware of that. So yeah, so this is the key. Compare ends provide the key idea of how to approach the problem. This is variation that true varies in, but dupes. The insert lengths are sort of natural variations of that type, which come along with every library. Now, if you didn't do paired end sequencing, you don't have that information. So that's a problem, right? So I mean, if you're doing single end sequencing, you don't know anything. You didn't get the version to it. So you can't always do it, but you know, it is there, and it is observable if you did sequence it paired end. Then it sort of was free. It was natural and already there. So you didn't have to do anything different protocol as long as you did it paired end. The idea of UMI. And so why don't we introduce intentional variation to attack this problem? And a natural place to put it is in the indices, because then they don't interact with the main trees at all. Right? It's something that can happen during the library that happens with the Daphne, doesn't interact with the main reads at all, so it's kind of like easily insertable into most protocols. So that's the idea for UMIs. So now that I1 and I2 reads can be bombed, right? I mean, in the days when there were 6 merges or something, there's no room to put a UMI. You need all those basic figures. But these days, index spaces are so long that we can commit some of them to the debug space, and then we got ones left over to do anything we like. And so let's make them UMI. And what that means is that those other positions in the index, we will arrange to be randomized. But when the guy that makes your adapter synthesizes them, you know, when you order adapters, you know that's for randomized basis, right? Because the chemistry is an incorporation process. If you're making a controlled oligo, then when it comes time, you're on the 4th base, it's time to come on the 5th base, and the sample the guy's orders the 5th base for oligo number 12 needs to be a C. Well, then you take your little tube of 3 Cs or whatever, it's only Cs, and you add that to your tube and you incorporate C. But the guy could also order it in. What that means is that you have a tube that's got all the nucleotides in it. And then when you put that on it with the terminator, individual copies in your tube will incorporate a random base. So now your tube becomes a mixture of sequences that are variable at that position. Right? Ideally, it's 25% ACP independently ID across all the ends. Right? So if I ask for 5 ends in a row, then that's 5 bases ACP, which is 4 to the 5th power possibility, 2 to the 10th power possibility, which is 1024. Okay, so ideally, if I order some oligo with the n 5 ends in it, I have 1024 copies in my final tube, all of equal representation. That's an idea that can happen, but we might not have to have that idea to be effective. We just need lots of variation. But that's what we do. We'll use sort of the uncommitted extra index bases that we don't necessarily need to be much. We will randomize them with ends. And then when you do the putting them on your library, when you put them on your file material, essentially that uniquely marks each molecule for your biosynthesis right at the beginning. Now, you probably don't have enough UMIs to really make them all distinct. Right? How many biomolecules did you pull out? Well, if you're in a high, if you're not certain for material, some fraction of Avogadro number, that's the possibility you have. It's just absolutely not enormous. And what's a long UMI? And we can buy that's 2 to 20. That's a million. So you only have a million labels for them go digitally biomolecules. So that's there will be duplicates. But when it comes time to do the dedupe with UMI, we're not comparing everybody to everybody. Because you got to pass the filter of how the main ends match first, right? You got to have main end 1 match. If you took paired end, that's Gardner. And then we're talking about adding extra filters that the UMI's got to match. But the UMI only really has to defend against the level that the other things aren't going to be able to defend. And so the UMI doesn't need to be 100 days long. So that's the idea. So that this will enable high fidelity debugging, meaning we keep truths. Every true that's going to, cases that we used to have as ambiguous, where we weren't sure what to do. Well, the sequence is matched. Looks like a dupe, but we're still not sure 100% whether that's really true or a false. But now we add this UMI on there, and the guys that came from different biomolecules almost, with million to one chances of the UMI member. And the high quality UMI, lots of diversity. We got a million to one chance now that the UMIs will provide. So if we ask that the main sequences and indices, which include the UMI, essentially, index if the DMUX part of the index sequences differs, the return different DMUX file. That's of course the example. So essentially, when I say main read and then index read, right. So, if the main reads match, then the UMI almost surely are going to come to the problem. Which subset of those belong together, right? So in terms of this problem of having a dupe group of 1,000 guys that all have the same main sequences, and then you look at the UMIs and cluster those, and you realize, oh, I've got 53 copies of one biomolecule, and we've got 147 of another, and then we've got the 12 of another, and whatever. You can tell exactly how many biomolecules you have, and then you just keep one read from each of those files. And now you have representation that reflects true biological. So that's the idea. So that's what makes ideal. UMI is sort of like the gold standard these days for dealing with duplication problems. Duplication problems of all types, platform and PCL. And a typical UMI size these days, maybe 8, 9, 11, a big one, 16 nucleotides, and so on. In practice, usually they're not perfectly diverse, I would say. They usually don't really, they're not like ACDT 25% for each uniformly across the whole. They don't need to be, right? They just need the property that duplicates from their fate keep them the same, which will be true. And that the biological ones that come from independent molecules have a high probability of being different. And as long as they can have biases in the UMI, as long as the biases are crazy, they're effectively well, they will serve to do the job. And furthermore, they would be very graceful. Right? In the worst case, if the UMI completely fails and every read in the whole line calls the same UMI sequence, you're just back to the situation you were before you had. They start there and do better. All right, so it's going to be beyond our scope to really get into the mechanics of how you do that and actually use them to do their D-group job. Instead, because they are part of index group, but I just want to just how we get them into their own files and sort of deal with them and Linux and so on. And it's pretty simple. We kind of talked about a little bit briefly in various points in the first few days. First of all, the formats that we usually see, I told you the lengths, but I didn't tell you where they usually occur. The most popular form we see them in right now is the second read being 24 nucleotides long, and it's entirely a UMI, or maybe some bases don't change in there, but effectively none of it is used for DMUX. That's one way to do it. You can commit an entire index read, right? Because we like things like unique duals. Maybe that's not the best way. Maybe you should have a unique dual and then still have a UMI. That's one. The next most common one is to have the I1 B16 nucleotide with the, one of our most common index formats is paired 8-8. That's very common. So increase I1 to 16 with the first 8 being as the 8 you had before, and with the back 8 being that's a very popular format. Well, these are rare things that we've seen once or twice or three times. So these popular sort of we've seen I1 is a 19 nucleotides, where it's 8 plus 11. 11 being UMI. I2 is 17, where it's 8 plus 9 with back 9 UMI. And we've seen I1 is 17. It's pretty simple. When you get to B cell capacity 2, all these things are basically just vector U base. So if you're doing non-demux, it doesn't really matter what happens to them, because you're not B cell capacity 2 doesn't have to decide any demux. So typically you can just keep them as part of the I index read. So in this case, you would have a I24 or something, and then you would get an I, I2. You get an I2 file with second index reads of 24 1 with the UMI read. But when you get to demux, then this is really different. Because in demux, the UMI bases do not participate in the of the demux file. You don't want to do that. You don't want to have to listen to sample sheet every possible UMI sequence variation. Inside the pilot that you felt BCL capacity 2 was probably put it in. No, you just want to ignore the UMI base. Before you're doing UMI. And so the way that is done is instead of putting them in the I, you put them in the Y. So if you used to have, if you had let's take one of these that's half and half. So the second one is I16, which is 8 plus 8 UMI. So at the non-demux stage, you might have had that as I16. Well, now at the demux stage, that has to turn into I8, Y8. So that means that the first 8 is an I, and it's participant, then that's part of how the demux index is put together. And then that Y8 on the back now says, well, don't throw those bases away, but treat them as another main map, like a virtual main map. And so at that point, if you did paradigm sequences, you might have R1, R2, and R2 files. R1 files would be the long 50 bits main end, R1, single line. The R2 files would be, in this case, 8 base UMI, which is coming from I1. Then the R3 files would be the paradigm. So that's how UMI is kind of look. Once you've got the UMIs in capacity files, how you use them depends on the tool. That's just how the mechanics, how they move around in the raw run folder. And since it's a demux workshop, that's what got them ready to go for the next handoff. Next stage. So typical UMI would be written down something like this, like the format for if you had this is actually one of the kind of standard image pairs. This is an I1 and this is an I2 and an 8 plus 8 plus. When people say UMIs, they often write it kind of like this. So if you have a case where it's I1 is 8 plus 11 UMIs to make it 19, and I2 is an 8, then people often write things like this, where the ends, here I use lowercase, whatever. When you see this kind of thing in indices, the ends are where somebody's trying to tell you what the UMI is. Okay. So that actually brings us to mixed index formats. So about 5% of lanes we see actually have mixes in them where people have combined libraries that, if they weren't combined, would have been run on different core cell formats. So that means like single indexing mixed in with dual indexing, or UMI mixed in with non-UMI. So how do you deal with that? For example, the documentation they have for their kits may not describe. For the single index guys, it's not going to tell you an answer. Why? It's a single index library. Right? You're running it on a single index. No, not when it's mixed in the paradigm. Now, so one thing people do at that point, which I don't recommend, I really don't. But some people then mix multiple samples. run these other captures multiple times. Use basis max there. So they would do the single and they would make a snap sheet with a single and sample. Tell these other captures to just ignore the equivalent captures. And they would do another batch with anyway. Kind of as one of the things I think I talked about in day 2 or something is that if you change, you really don't want to really consider lanes in smaller pieces than really exist, especially if you change the basis, you're going to pay attention to make the decisions about what piles well fit into. It's much more logically clean, and describing what situations the piecemeal approach can actually kind of work decently and when it doesn't is complicated. And what I recommend instead is simply doing what I call either lifting or altering the situation so that you can run it in a single shot in the one B subtask that you run and beam up everybody all together in some logically consistent setup. So what does that mean? So the lifting is relatively easy. So lifting means that if you have first of all, physically, of course, you have to run the run to the maximum of government, which has already been true always. But a flow cell is more than one lane. So if you put different people's lanes together in a flow cell, and this person asked for 58, 850, this person asked for 50, 10, 10, 50, then obviously physically you got to run 50, 10, 10, 50. The guy that asked for 80 is going to get 2 extra I1 cycles and extra I2 cycles, which then, if he's not in this particular, you can simply ignore the thing. I mean, that's the purpose of the more cycles and use basis map. But there's no issue because it's a lane 1 versus lane 2. Here we're talking about mixtures of these symbols. Anyway, yeah, physically the run of course has to have enough cycles in SCI when I do to cover the maximum that everybody is participating in. That's a non-negotiable. The question is what to do now that you've kind of collected more cycles and some of the samples are nominally known to be. I've already told you the empirical approach. And furthermore, you can kind of guess, well, what are these indexes coming from? Well, the adapters. And these adapters don't, it's not like if you try to sequence past the end of the sequence in the documentation, this sequence explodes. No, you're just going to sequence the next base in the adapter. And pretty much the next base in the adapter is going to be something stable because people don't usually when they synthesize all the insert extraneous unwanted variation, right? I mean, the index read is variable here. They're not going to design the surrounding parts that have more variation. And if you actually get to the point where you run off the adapter, so that the sequence goes dark, dark is poly G. So even if you run off the adapter, you at least get stable. So the upshot, or or if it doesn't prime at all. So the upshot is basically that no matter what sequence of documentation tells you it's going to sequence for a particular get to a particular length. If you run that longer, you get something that continues stable. Now you don't know what that is, maybe, but you know the empirical approach, and you can simply find out. You go to the empirical histogram. You've got I8, but your documentation, you're the I8 guy on this list. Your documentation is the 8. You got mixed in with this guy that's got I10, and you don't know what the 9th and 10th bases of your indices. No big deal. Go to the empirical history. We'll find the matches to your guys' first 8, 9th and 10th bases. You can very easily, it's usually so stable that it's not really a big deal at all. You can very easily extend your documentation to 8 or 10. So in that case, you can make a single sample piece of all 10 team up to in one shot. You can also, I suppose, take the 10 guys and just throw away the 9th and 10th base. That's a stupid way. Obviously, the less bases you use to make the decision, the more likely it is that sample is live. Why would you do that, right? You want to lift up the more biggest guys to the nicer level, the longer so that's the idea. And pretty much all situations like this kind of it's not really a big deal. So you may have single index libraries, but they get to the parent guys. You don't know what the I2 is for the single guy. Just go look in the empirical. may all be the same. That's common. In fact, it's usually something about y-axis, y-axis is the fastest relation to the singular guy. Five comes to your sequence that is and the adapter is standardizing that. It's part of it. When it was the phone turnovers and the Nova sequence, nobody wants to know anymore. That's all I'm saying. I'm not saying you should like just do it once and forever. Assume that the extension is always the same. You start it to the first step. Ideally all the time. But yeah, if you're going to single line guys, you just go and you're running them with a paired guy, you just go with the interpret pair. Very obvious. This is going to make the paired example speak to how it sends everybody to the paired. So maybe a slightly more confusing situation is the UMIs. So some samples have UMIs, some don't. The easy case is if the UMI is in cycles that none of the non-UMI guys use. For example, all of your non-UMI guys are first index 8 bases. The guy with UMIs is first index 8 bases and then UMI. Well, those UMI bases then only matter for UMI guys. They never collide with the demux bases of the other guys. You can simply pretend that the non-UMI guys have a UMI after them. You're going to get some sequence there. It's going to show up in R files, or R2 files, or whatever. And then they're just not usual files. They'll have sequences. There's no need to not. BCL Fast doesn't have syntax that says some samples get this and some not. Whatever, you just make some extra files if you're not going to do it. The slightly more complicated case is what happens if the samples of UMI bases have those UMI bases on bases that the other samples want to use UMI. That's a little tricky. In particular, that can be tricky because now UMI bases are random. And they can, with some frequency, match anything. So now there are some cases where you can argue that the distribution of values in the UMI and a particular person the confidence chance is low, so the mismatch chance is low. I generally prefer to like not have to think about that and worry about that too much. So the nicer situation that is in that case is if you took the UMI, if you take the bases that are UMIs in any sample that is and throw those away from demux from all the other samples, so that the demux is done purely on bases that are not UMI. Hopefully, at that point, the samples are. And you could get unlucky at that point. It's really good in a world where it matters like what the distribution of UMI probably is. Much better ahead of time to design the mix so that that does not happen. I mean, sort of these mixes of this format, sort of, I wouldn't recommend them, but they happen because whatever people have problems. Yeah, so I wouldn't recommend mixed formats in general, but once you're in mixed formats, you should, if the UMI is involved, you should check that. If I throw away the UMI bases for a lot of my non-UMI guys, do I still have enough uniqueness to keep up? Assuming you do, then that's simply what you do. So, for example, if it's 8 plus 8, I 1 and 2, or 1, 2, and then the other samples are 8 plus 8 UMI in that one, and then nothing plus that's 2. Then what do? Well, first problem is that these samples have an I2 8, and this guy has an I2 0. That's okay. Go to your empirical histogram for the pairs, start looking up the guys in the UMI file, and find out what their I2 is. So then you can convert the UMI guys from 8 plus 8 I1 plus 0 I2 to 8 I2. Well, now the second index is completely the same. So now we focus on the first. Well, now we're in the situation where this guy's got UMI and this guy's not, but the debug cycles are the same 8 cycles. So then you just upgrade this guy to have 8 plus 8 UMI. You're just going to ignore those basic files. this lane becomes 8 plus 8 UMI I1 plus 8 I2 for everybody. Yeah, so the case I'm talking about here now, so these examples, these are the lift examples where everybody's upgradable to the same situation. The ones I was just talking about are what I call the alter case. And that's where you've got to downgrade somebody basically to the top. So like if it's 10 plus 10 for the non-UMI guys, 8 plus 24 UMI for the UMI guys, you can lift the I1 8 to 10, no problem. That'll make it very good. So then it's 10 plus 10 versus 10 plus 24 UMI. But now you've got this 10 here, which is DMUX 10 versus 24 here, which is UMI 10. That's a problem. But this sequence can create matches and stuff here, and now you're worried. Then you got to sort of check it. Is the I1 going to save you or not? Well, hopefully, right? Because if these share I1s, well, now you're down to like random chance that UMI is going to get one of these guys. So that's not to get yourself in that situation at all. So hopefully at this point, the I1s alone are enough. And that's what I would recommend. So we upgraded this to 10 plus 24 UMI. This is 10 plus 10. This 24 UMI cycle completely kills this guy. So basically, this becomes a 10 plus 0 or a 10 plus 24 UMI. And this guy's a 10 plus 24 UMI. 10 plus 24 UMI. That's what I call it. So yeah. Yes. So in that case, many of the things that we've talked about, you sort of don't know. You've been mixed with people you have no control over and no knowledge of. And I just, if it was my personal lanes, I would try to avoid that situation because there are effects from it. I would rather if I can't put together enough to sequence the whole lane myself, I would rather find local people that I know can put together something for that. From the company's point of view, I have a full lane to look at. That would be the way I would try to do it. But of course, the advantage of that situation is the company is able to take from bigger pool of possible collaborators. But yeah, but that kind of model for operation has that you may not even be able to really completely figure out. If you're not going to have it. You can have all these things in this popping, all kinds of things. You can have other guys team up with their files at rates. Maybe if they're in mouse and you're in the elegant or something, you're going to know because you do your alignments. 5% of you still align. But one of the things you should do in an alignment-based pipeline is have a fairly large pile of unaligned. Like randomly sample 10 leads out of there, put them in the NCBI web flat. And you'll find out, oh, well, they're like all mouse. Well, they're not mine. But yeah, you know, like the guys saying, well, this is you. I mean, it's just not for all the things we talked about in these 3 days, you need to appreciate the fact that's not risk. So I wouldn't recommend this format where you, okay, well, I mean, you don't always have full freedom, right? Ideal is you need to deal with you. That's sort of not, can't always have it. And so this is a case where the UMI kill by group. And, you know, regardless of whether it's mixed in with a guy like this or something, where you can actually kind of like this guy and turn him into a single. But I don't, you know, if you have your choice, I wouldn't do a UMI this way. I would arrange for the UMI to be a piece of I1 or I2 or possibly control, retain dual indexing rather than give up dual indexing. Because the dual indexing is your defense against index hopping, and the UMI is your defense against two. Rather than both. So in that case, it's less of a problem mixed, because as long as this guy is 8 UMI or something, this guy's 10+10, then we'd only have to kill the back two bases. Right? This guy's UMI would start with the 9th base. So then we worry about the conflict of 9. This guy, maybe we consider it 10+8. And consider this guy 10+8. All right, so that's an hour, so we can take a short break there. Yeah, that's a good place for it. Well, you're probably unless you can protocol at all. So, that comes with all the reagents, very detailed stuff. All the interface between your sample and the kit is defined. Axionograms of size and maybe the input fully marked. They will have to find your job is to get your raw material to find input. That's the life. That's the kit maker. Unless they define what they're doing. As a business, depends on whether their internal philosophy is focused on what. Are they focused on turnaround time? Are they focused on quality? If they're in a situation where they need to put together a run, and come in for a while, and you put that two business to another guy, it may put together and have a power. I don't know, right? You have to see what the service is. Yeah, I really recommend for mixers that you a priority check to be this together. But if the company is putting it together, have no control. So you could ask them, like, you guarantee that all of the users in the name on the rule, you're not going to mix in single. Are you going to not format together? You have to ask them. That could be another guy's sample. At least at the cores at UCLA, you'll generally be able to find out who's on your lane. If you're with BSCRC, it's because most of the object of transaction is the lane. So if you're next, it's because you figured out who to mix with already. JCCC has what probably company the extension for things, and I close it everything out. Index hopping, big color, don't know. You don't have any way to even guess it. I mean, you can sometimes estimate because of things like a line spiral. But that's how that happens. Things we've talked about now that you appreciate how that happens. In general, I would really resist random. You just don't know. As I was saying, one of the days, it's not like the process. Adding things convert. Adding things doesn't just do convert. Adding things can actively damage. And so that means you need to track the process. Companies doing multiple units together without all your trust in them and all your trust in everybody that's contributing to that. Okay. All right, so yeah, this is just going to be one slide on sort of like essentially this summarizes all the problems that I see. You know, we could oh, screen sharing got lost. No, I think it's still on. You are screen sharing. Yeah. Okay. Okay. So rather than kind of really take a lot of time to kind of do examples for all these and so on, you can at least, I mean, I'm sort of arguing for authority because I haven't shown you the evidence. I was saying that I've actually demuxed these all 600 lanes and I've seen these things and so on, but if I had to, I consider the real evidence and so on. But maybe from the procedure of empirical discovery that we kind of showed you the outlines of the first day, you can at least believe how you can come to the point of discovering these things and so on. So here's basically are the major classes. So first one is if the user sample sheet is wrong in some pretty straightforward way, or could be a pretty gross way. So it could be that, for example, all of their I2 sequences are reverse copies. You think, well, how does that? Well, it's because whether you put a sequence or a reverse complement there is related to the platform. So if you're on Novaseek version 1.5, it may be one thing, but if you're on an iSeq, an iSeq, or whatever, it's different. And your documentation kit for as long as it's not a really old kit, your documentation kit will actually have multiple columns for on the Illumina platform. I don't think the I1 has ever. The I2 has this problem. Some of the platforms do it one way, some of them do it. And the sample sheet will then generally have 3 columns. The I1 column, but then there's 2 I2 columns, one of which is the first column. And you pick the right one, the one that's later. If the sequencing has Novaseek, you need to know your team to put the right sequence on because it does depend on. And because there's so many columns sometimes in those files, sometimes you get things where like some manufacturers will give you 2 columns, actually 4 columns. They'll give you the I1, I2 for one set of machines, and then the I1, I2 for another set of machines. Those 2 I1 columns are exactly the same. But some people get the columns mixed up and they'll somehow they'll give us 2 copies of I1. But you may have a sample sheet where it's 2 indexes with the same sequence down, anyway, things like this. Just be careful when you're making your sample sheet if you're actually reading the text, following, knowing the platform you're on, actually putting the right sequences. Sort of simple. That happens to surprisingly number of times. And one of the first things Shua and I ever did was demux columns. I mean, those are usually quite obvious because it's like total reads. Demux tail percent would be 9%, 8%, 9%, basically then we know already. Well, we should try reverse calculating things, flipping I1 and I2. You do those permutations, sometimes you find it. But that happens. That's the whole class, you know, that's a major class. Big one is the violation of the mixing rule, by which mostly I mean things less than 2 dimensions apart. So mixed components that are 2 dimensions apart, even when that can be sort of what I would call intentional, which is people didn't check ahead of time. And it's like you had your job responsibility as a submitter is to make sure that things are far apart. So your job is check ahead of time. Sometimes what trips people up though is that there are mixed components that they're not expecting. So when we were selecting phi x, this is why. So phi x has polygon on the front, I1, and it has this particular sequence for I2. So if you don't check when you're doing your checks for distances, if you don't check against that sequence, that guy can land close to you. And that includes both the poly G, because some kits include indices that are like 6 out of 8 bases of G. I wouldn't have done that, but you know, whatever, they're out there. And that means that will always be a two mismatch out of phi. You figure its spikes are so common that libraries, anyone making kits with this, like, why don't I just not use that one? Especially if it's a likely concept. You know, they're out there. And also with the I2 for phi x. So people may have checked, but they didn't check the fullness. And so I would say you should always assume that the phi x sequences are in there, even if they're not quite well, avoid it. You also need to avoid I2 polygon. That goes up a lot. And you need to more rarely reference using some level of I1 polygon. Just avoid all those stuff. That's a big one. And it is a major source of trouble. Especially with somebody, I don't know how it happens, but they'll get 2 targets and 2 different samples on this one mismatch. So you know they're going to tell it. You've seen how one mismatch is often needed to recover one library without another. There's one mismatch of the intention target to target. Those are people. You're asking for another major class is essentially missing samples and extra samples. Missing samples are more obvious because you may have decent representation on close schema for a bunch of guys, and other guys are so low they're essentially 0. So they look like missing values. Well, now you have to, hopefully then they pay attention to the Emux fail, and if Emux fail is big, then that's probably where they are. But it means your sample should be part of it. But you know, if it's a really big mix, like a mix of 100, only 2 are missing. Emux fail can't really tell you whether they're there. It's just too small to vary. Especially if the lane representation varies. So those are create lots of but, you know, oftentimes you can recover them because they're sitting there. So the empirical approach is very good at finding. The empirical approach, you let the lane tell you what's really there. Missing guys will either be kind of depending on how low you want to go in the histogram. If you try to know that they're missing, you can dig further and so on. For example, if you know what box kit they use, you can try looking for other box sequences in there. Maybe they make a mistake. That way, this depends on your experience with how well you are sort of dealing with the histograms and how far the baseball errors in the length can fuzzier or sharper. Anyway, the missing ones can often be found. If it's just a really straightforward case where the representation isn't out there, it's really good. You made a mistake and you put on sequence or on the website, or something. It'll be very clear, right? They'll have a sequence that is not essentially very including this one. But then there's a super bright guy that's near the top of the histogram that's the screen, the IMA sample, and then if you look them up, it's the standard sequence or something off. And so clearly, somebody made a mistake and used the wrong 2 with the level wrong or something. Now, you always worry in those cases because that's really sample thing would be the big, but no, but a lot of the times if you've got a mix of 20 with one missing as a 0 and extra diet representation with the others, the one missing diet is that one extra diet. Probably only like more than like. But then you get cases where people are missing 12 and you've got 6 extra. And then, well, it's not really your job as a Unix guy to sort of tell them what to match up, but at least you can kind of say that's the situation. You got 20, 12, or you asked for 20, you thought you had 20, you got 12, you're missing 8. I found these 4 extras that are really, really there. They're clearly extra. I look hard, there aren't really 4 more, so that's the situation. You thought you had 20, you got 12, you're missing 8, you got 4 extra. But yeah, you get cases with 1 missing, 1 extra, 1 missing, 3 extra, 12 missing, 6 extra. Sometimes you get 6 missing, 8 extra. Yeah, whatever. I mean, it's just things happen, but resolving those cases in terms of recovering samples, that can be tough. But at least you can find information that helps try to figure out. And another big one that really creates problems is the high representation. The lanes where you've got 2 500 million guys, maybe that's 1 project, 2 projects. It's a multi-project mix. Multi-project mixes is very common. 3, 4, 5 projects together. By project, I mean one lab of RNA sequence, the lab of very, very common. Maybe one of those is a 2-plex high representation, and the other one that's in is a 50-plex low representation. And so that makes the sort of the 500 million guys so crazy high compared to the 50-plex. So the 2 guys that's not maybe 2/3 of the lane already or more. The 50 guys that are left are fighting over the scrap, and so the ratio between them and the top guys can be 100 to 1, 200 to 1. So that exacerbates all the problems with the top. Right? So then you're asking for base colors going into a place that complexity of industries is compromised. If the bright guy, the 500 million guy, is sequence close in indices, the ones that are low guys, it's the base call error right alone. Because he's big, a little bit of bleed here, big contamination level. Yeah, there's lots and lots of trouble. Normally, one of these things you can kind of survive, but like if you're kind of got one of the other problems and this thing, then you can great. So yeah, then another class is you can get cases where the systematic base call corruption of a guy. We saw cases on the first day where like 30% of the representation can be lost. But the example I think we used to have the variant where actually the expecting her was still the top one, and then a little bit lower down, 5 variant guys, but he was still below. So you can get where the top sequence for a guy is actually what looks like a problem. You kind of talked about that in the update. The top one. At that point, you want to do something to capture the top one. How do you achieve that? You want to capture that guy that people didn't expect it actually really. Some other class of problems are all derived from the usually high mismatch window rates in the unit signals, usually triggered by something else with the higher. Or you do something like, suppose you're mixing single end libraries and dual libraries. Now, the sizes of those two on a sample by sample base may be comparable. Maybe this is a 10-plex and this is a 10-plex, and everybody's representation is probably. But the I2 for everybody in the single index guide is all constant. So when you compare the I2 representation ratio, the fixed I2 here is 10 times, all 10 guys here just combine their power, right? So they become half of the length. So the all these folders, 20 parts, and 10 parts are all the same. So this guy is now half. So if you half of all the I2 repeats the time to do the same sequence. That may even be sort of survivable, but then just make these guys a little higher representation. These guys are bigger flex. These guys are situations where no, nominally there are 100 different samples in the range, but 98% of the I2s are trying to do a function. So in that case, even if they thought the I2s are crazy, the guys that rely on I2 to actually do something are not going to be happy. It's because the single index guys are good. So yeah, so that kind of thing can be a trigger. We talked about index hopping. That's another class of problem. So sort of a gallery of events. And you can get very scavengers for the bias, but talk about it. But that was basically the realization that this happens with and then what's really the nightmare is when like web links have all, especially high rep ratio with the close one, or high rep ratio with the mismatch one, the high baseball areas. Those become very hard to resolve because essentially what you're saying is that the critical histogram is very hard to evaluate whether any given sequence is a new guy or not, or something. So it's really you start combining them, you can kind of go that fast with the interpretation of things from a very important. Okay, so basically that leads us to the summary of things to do and not do, which at some point I'm kind of repeating myself over and over, but I hope it kind of fits. So it's just basically all the things that we talked about today, collected into either don't do these things or do these. We've talked about them all basically, so I'm not going to leave them again. Just 20 minutes talking about some of them right now. So I'll just leave this slide for you guys to read on your own time and whatever. And show this to your friends or whatever. I mean, but this is basically the summary list of like this is the story from what I basically the summary of stuff I've learned. Now I can wish for perfect situation where you really don't do any of the dumb things, you do all the things. But it's not like a binary thing, right? Some of these things are easier to do than others. You do the ones you can, avoid the ones you can. Okay, and so the last thing I want to talk about is custom indices, and actually give you some evidence of how the stock fits really do have some issues. So one of our myths from day one was that the commercial prep kits are well designed and optimized in terms of their index tag, and that if you mix things, that the mix conflicts. And even if you think, well, okay, I'll stay within one manufacturer, I'll be fine. You may even think I'll stay within one box, I'll be fine. That ends up being false too. So here's an example. Illumina has quite a few systems of indices. The original batch for the modern machines is the true batch. They also have unique tool indices. That's their unique tool, the UDI, as they call it. And there's a thing that's the 96 box. The 96 box, which actually has two secret versions. And there's two catalog numbers, and there's like a couple indices that change sequence between the version numbers. And we've had people put the wrong version numbers on the sample sheet. And it's really tricky for them to figure out, because I think what they told me is that when you are in the lab and you have the box, the physical box with the physical, the catalog number isn't on the box. So then you have to go look at your order sheet and find out what you ordered. It's like the only evidence you have about what version it is. That ends up being like when you basically have a missing sample. Empirically, you have one missing sample and one extra sample. But then it's kind of really easy. You go then and ask them, well, what sequence is the missing sample? It's UDI 55, E1. What sequence is the extra sample? It's UDI 55, E2. So that's just very clear. Anyway, you could have asked for a little better labeling and documentation. Anyway, so that's why like There's a 16B2 in that section. It's also 16B1. And then they have the Nextera system, which the ones I've shown here are part of the Nextera non-unique. And Nextera bought like computer makers, so originally developed outside of it. But anyway, but it's only the Illumina broadly now. If you didn't know the history, you wouldn't know that it was a product. So that's 3 systems of Indices. They're all 8 merged, actually. Well, the true seats you could argue with 6. Call them all 8. And conflicts only get less if you make things longer anyway. So here's 3 systems. I'm not putting all the indices here, but I'm putting the ones I'm putting here because I can't even remember if these are really all the conflicts among this set of boxes, but it doesn't matter. It's not trying to shoot conflicts. And so it's a subset of true seats that has a subset of UDIs, a subset of the Nextera. These are the I1 pictures. So the next, this is dual and this is dual, but single. But I'm showing you I1. And what do you see? Well, the red box is actually the projector doesn't make it very clear the difference here. You've got the slide there. So these red boxes, which are one mismatch, and there are red boxes. And you got the orange boxes. I probably could. The orange boxes with the 2. And both of those, let's say, for present purposes, really are 2 colors. Because once you go below 3 mismatches, remember that means that becomes now possible to have index trees that are 1 mismatch away and equidistant with 2 guys. And if you show SP2 will no longer allow you to do up this way. Numerically, that's not a good 2 mismatches may be survivable sometimes, but 1 mismatch is you really never want to get a situation. And what do you see? Well, of course, within each box, it's fine. But if you cross these boxes, they're not fine. And so there goes that. So, and here's the I2 picture. The true seeks a single index, and when you do them as dual, they all collapse with 1 common I2, which is single 5. That's what is only 1 gray box on the left corner. The true seeks went to a common one. But then there's the UDIs, and then there's the Nexteras there. Now the Nexteras are fine within themselves of the I2. But even before we look at cross boxes, the crazy thing is that the Illumina UDI I2s themselves aren't part of it. It's the same box. Crazy. If you don't believe me, here's the web link. You can go to the page and that's documentation. I'm giving you like some here. Compare UDI 23 to 5062. There's sequences up there in the top there, and it's 2 mismatches apart. Why? You can plug these into index checker yourself and generate this matrix, right? So if I grab this column, there we go, and I go to index checker, reload here, and then I'm going to paste that in. And what do you see? Well, there's the matrix where all the conflicts lighted up. What can I say at that point? So sometimes you're in a situation where you can actually design custom indices. Some people are good protocol development. Some companies actually like to order custom indices. But sometimes you have a chance to choose your tag. And that can be a nice chance because then you can actually, carefully avoid all the problems and make better ones. So for any given fixed tag length, any fixed chamber, any fixed length, and if you have a fixed error model where suppose you're trying to defend against one mismatch error. So if you're only concerned with one mismatch error, you know that as long as you stick to at least 3 mismatches apart on your tag, you have that one. So under those parameters, it then just becomes a combinatorial mathematical optimization problem to figure out how big you can flex. Obviously, you can always do a 1 flex. That's always compatible with itself, to any degree. And obviously, if you want to have a flex that's more than the number of tags, So if I'm doing Kmers and K is 10, then there's 4 to the power 10 possible ACGT sequences, which is 2 to the 20, which is a million. The binary, maybe or whatever, instead of mega, but basically it's a million. And so if you want to have a 2 million plex, you can't do it. This is not enough distinct frequency. I probably should have used a shorter length. If your tags are tumors, right, it's 2 letters, then it's only 16 of them. There's no way you could have 20. It's not enough. So clearly, between those two extremes, somewhere there's the line, right? Somewhere there's the last time you can meet your error model before and still do it with your length, and then the next bigger size is not possible. And so it's a mathematical chemical optimization problem, not a particularly easy one in the general case to find that largest plex in the now you can have other constraints and so on, like are there low plexes, all low plexes subsets also different. That's not a situation that really even applies so much. Once you're worried about the moderate pi plexes, sort of index diversity, sort of automatic index diversity problems right now mostly all derived from representation ratio, which is effectively decreased with some guys. Usually that's a much bigger problem than whether the sort of the equal uniform the function about a small plex is more like library size, which are not under control of the index design directly. So anyway, but can we then under some of these constraints find better sets than commercial? Well, yeah, you can. So I've done some of these optimization problems. I don't, for most cases, I do not actually know the precise maximum possible. I only have the highest size I've been able to find so far. It's really hard to prove that there's not. Anyway, so for example, if your concern is the one mismatch recovery, so the minimum parent distance of the tags within 3 mismatches, and you're willing to use 8 merge. Actually, I have a set in hand of 1,330 that are all mutually 3 plus apart. So that's a 1330 plex that meets that condition. Whereas clearly these Illumina sets don't meet that condition. They're much smaller than 1330 and they're having 2 and 1 problem. So they don't have to be like. In fact, for this condition, if you go down to 6 merge, I can still give you 164. And they still meet that product. So the 8 merge are kind of overkill for that you're only sizing like this. In fact, if you're at 8 merge, you probably don't need that size. You can actually either bump up the mismatch tolerance, or you can actually start to even mix them around for indel. Anyway, so if you're in this chance to design custom images, I recommend yourself basically an expert on this process, because we can probably give you some better sets of indices than you might think are otherwise possible. So here's just a put the money in our mouth here. So here is the list of 164 6 merge that say at least part. And I can copy and paste this list, and you can copy and paste this list. More importantly, you can either put write up a little R or doing something. I don't know if you want to trust the web tool or not, but basically, there's enough information for you to verify. You've got the sequences. You can verify that every one of these to every one of these other guys is always at least 3 mismatches apart. I don't know what happened there. You can see the web tool takes a little time to think about that one. That's a 164 by 164 matrix. Or maybe Firefox is the one that's trying to figure out how to draw that. But anyway, there you go. I put at the end of each row the summary of the minimum distance, and you can see they're all basically 3s. And in fact, actually, at the beginning of the output here, it tells you the entire range of the whole matrix is 3 to 6. So 3 is the minimum. Yeah, as opposed to, let's take Illumina. Forget even the multi-box stuff. Let's just take TrueSeq 1 to 48. That's like the original set of 6 merge. All right, we'll put them in there. And what happens? Gosh, there's 2s in here, right? So not a lot of 2s, but there are pairs of 2s. So that is a set of only 48 that doesn't meet that condition. Right? Whereas this is a set of 164 that does. And so these two shrunken versions of that conflict matrix, where 3 pluses of white and the 1 is a red and 2 is an orange, these are to scale. So this is 48 versus 164 is the size ratio. It gives you some sense of the scale. And this guy's got these 2 conflict bases, and this guy has none. So that's when I say you can do better, you can do better. And so maybe that takes us to kind of what would be the kind of the ultimate indexing. So, I said we got 36 bases we can basically play with. So let's use them. Let's maybe do unique dual, of course, with UMI. Maybe even unique dual with dual UMI. 13 plus 5 UMI for I1, 13 plus 5 I2. That would give us a 10 mer UMI, 1,000,000 to 1 resistance beyond sequence matching of the main ends. And then we have a 13 and 13 index, which is a decent length. And we, depending on, it depends on how much of a plexity you want, you have choices there. But basically with 13 mers and for typical plexities, 10, 20, 100, 200, we can make those far apart from each other, far bigger distances than are typically existing in kit boxes. Right? Way more mismatches and also keep indels apart. And so, and then BCL the fast 2 isn't going to really be able to do a good job of demultiplexing those because its model is too small, too inflexible, and the mismatch error is too inflexible. You need to do it from custom defects. But even if you didn't do the custom defects, the custom defects benefit is the fact that you can do very good recovery in the face of pretty high base call errors on the instrument without making a lot of misdemarks, base call errors or misdemarks. But even if you were to just use BCL fast 2 and give up on sort of the high recovery aspect of that, you're still going to get the high resistance base call errors because the indices are so far apart. And furthermore, it's sort of like, okay, the last thing defending it, we've got the dual work defending the other thing the last thing that's sort of hard to defend against is what happens when one sample, the sample, the representation is very high. Can we defend against that? In the extreme case, what happens is one sample like takes up the whole range. Right? Well, how do I force diversity if I've only got one index left, right? If one guy grows to take over the lane and squeezes everybody else down there, how do I have an index diversity? Well, the answer is that because the indices are so long now, the complexity can be quite high. And so I can actually afford to commit multiple indices to every library. So maybe you want a 96 plex. Well, we'll design a 384 plex, and we'll give each of your guys 4 of them. Now, that's not really much more work. It's more work for the oligo maker. It's not much more work on the wetband stuff, because when you have the tube that's got adapter 27 put on your sample, you don't care if all the adapters in there are the same or not. With the UMIs, you don't care either. It's just mixing, it's just having a bit of a mix, not only with the UMIs, but also with the tag. It's not going to be a continuous end-to-end mix of UMIs. It's going to have a few discrete copies of it, 4 choices that are very far apart. So but then basically, even if the rest of the samples in the lane were to completely fail, and this one guy took over the lane, he's going to left, the indices are still better because he kind of acts like a little 4-plex all by himself. In terms of demux, if you're doing custom demux, you put them all in the same output file. If you go BCL fast 2, you've got 4 target lines. You just combine the files. And that then basically gives you sort of like the best world out of anything we've talked about. You've got high recovery, high resistance based on errors on the machine. You've got index hopping resistance. You've got dupe resistance. You can do large plexes. But I don't have anybody that's taking me up on the offer to do one of these yet. So we'll see. We'll have to see the first time that we're able to run one of these, whether we can meet all those a priori assumptions. This is sort of the idea of what sort of like indexing could be, and it's achievable now. Okay, so that's it. Hopefully this is useful to you. If you want to give me feedback, that'd be great, because this is the first time this workshop has existed. Things that work, things that don't work, whatever. That'd be great. So I'm going to stop the recording here. And then we'll take questions.